Assalamu alaikum girls, this is your biology class and we are going to start biotechnology chapter number 17. Biotechnology, it is actually the relation between biology and technology. So something which is made up of biology and technology both is known as biotechnology, and that is what you have already known. Like, what is biotechnology? We are going to study about that and how it is going to prove favorable for the mankind, which is known as genetic engineering. The branch is genetic engineering, and we are going to study about single cell protein also. So as we know that humans have been using biotechnology since we have discovered farming and the plant growth, crop production, all these things have been possible because of biotechnology. So we have also studying animal breeding, and that is also a form of biotechnology. Cross pollination and cross breedings of animals actually were the main and major techniques of biotechnology, but these techniques were used because of these techniques we had product quality, and the product quality was increased. So we wanted to we wanted to gain the specific requirements, and that is how we went into the details of biotechnology and then started engineering the genes of the different organisms, which is known as genetic engineering. And this is how it all started. Introduction of biotechnology, it is defined. Now please underline the definition. It is the use of living organism in processes for the manufacture of useful products or for services. So if we are going to use the living organisms for the production of useful products or for services also, we are going to actually uh, like do have like we are going to use experiments and different things, and that is known as biotechnology. And it can be breeding also, like cross pollination and interbreeding of different species also, but in the advanced state is known as biotechnology. Although the term biotechnology is new, the discipline is very old, very old. So the fermentation and the other such processes which are based on the natural capabilities of organisms are commonly considered as old. So that is what I was saying, that normal different processes, for example fermentation and interbreeding, is known as old biotechnology. But the new technology is the artificial synthesis, the modification, the removal and the addition and repair of the genetic material is considered as the modern biotechnology. Why do we do this? We do this to alter the characteristics of the organisms, and this started in 1944 when it was first discovered that the DNA carries the genetic information. It was always known, but it was proved in 1944, and then scientists they started isolating the enzymes of DNA synthesis. Why do we need these enzymes? To make a cut between the plasmids and the other DNAs, and then take out the genes of our interest, and then prepare DNA outside the cell. In 1970s, they were able to cut and paste the DNA of the organisms, and in 1978, they prepared human insulin by inserting the insulin gene in bacteria. So that as you already know, the bacteria started multiplying with the gene of insulin, and the insulin was produced in the bacteria. So human growth hormone was also synthesized in bacteria. Human Genome Project in 1990 was launched to all to map all the genes in human cell, and that is how the DNA test was possible. How? Because we have mapped the human genome, and then the changes in the genome are now seen relatively. Okay, so here it shows the 19, like the embryologist, he made a sheep which was known as Dolly from the body cell of an adult sheep, but obviously that experiment was not successful and that sheep died. Human began using microorganisms as early as 4000 BC for making wine, vinegar, cheese, yogurt, etc. Some of these processes have become a part of every home, and that we hesitate to refer them as biotechnology. Actually, it is biotechnology because we are using the living organisms for our betterment. For example, make for making vinegar and for making cheese, we put different bacteria which ferment our products, and then this happens as a result of biotechnology. In the recent years, like, biotechnology is a complete separate science. Why? Because it has so many different dimensions and so many different branches. It has attracted the attention of many intellectuals from diverse fields like agriculture and medicine, microbiology, organic chemistry. The scope for biotechnology is so wide that it is difficult to recognize the limits. The following are some areas of application of biotechnology. And biotechnology in the field of medicine. In the field of medicine, the biotechnology synthesized insulin and interferon, which is antiviral proteins, from bacteria and released for sale. A large amount of vaccines and antibodies, human growth hormone, and other medicines have also been produced. So, you must know that in the field of medicines, what are the services of biotechnology? Production of insulin, production of interferon, and many enzymes have also been produced. Gene therapy has also been there, and it is very important in recent years. Biotechnology has also proved beneficial in forensic medicine. The study of forensic means something which is related to all, like, operations and all these things. Okay, the study of DNA helps in the identification of forensic means related to criminals. Like, if we want to find out different criminals, so that is forensic science. Biotechnology in the field of food and agriculture, fermented foods, for example, pickled yogurts, malted foods. See, malted means powdered milks and all these different things. For example, malt drinks. You know what are malt drinks? You put them in uh, milk and then drink. So, wheat, barley, flour, and all these things, various vitamins and dairy products are produced by using microorganisms. Wine and beer are produced in beverage industry. Biotechnology has also revolutionized research activities in the area of agriculture. Transgenic organisms, these are organisms which have a different modified genetic setup. So many different transgenic medicines, transgenic organisms we have been taking. We have been eating all the different items which are present in the markets. All of them are mostly, but like, all of them are transgenic. Like, we have changed the genes. Why we have changed the genes? Sometimes to increase the the shelf life, because we want to put them in the shelf in the markets, and we don't want them to decay, and we don't want them contamination, want them to be contaminated. So we do what? We change the genes of these plants, and as a result we get the fruits of our choice. For example, the taste can be changed, the quality of fruit, the size of the fruit, all this is changed. And in which desirable characteristics are present. For example, more yield, which means more bigger size, and resistance against diseases. By again it changes the shelf life. If the disease resistance is more, the shelf life is more. Insects and herbicides. Now, this was related to the plant. Now, transgenic goats, chickens, cows, they are also present, and they give more food and milk, etc. Many animals like mice, goats, cows have been made transgenic to get medicines through their milk, blood, or urine. Again, the same thing. We want to multiply the medicine and multiply the chemical, so we are going to insert that gene into the gene of some other living organism, and then get our desired product. This is how it works. Now, biotechnology and environment is the next topic. Biotechnology is also being used for dealing with environmental issues like pollution control, development of renewable sources of energy, a restoration of degraded lands, and biodiversity conservation. Bacterial enzymes are used to treat sewage water to purify. Microbes are being developed to be used as biopesticides, biofertilizers, biosensors, etc. And so it means that some of are the pesticide chemicals are paired up with the any biological entity, which means any organism. So we call them biopesticides. Biofertilizer is the same thing. The fertilizers are inserted in any kind of living organism, so biofertilizers, and then biosensors also. Um, these are used for the recovery of metals, cleaning of spilled oils, and many other purposes. So, what are biosensors? Some of the chemicals will be present in some of the biological entities, for example, bacteria, and when these bacteria is used over spilled oils, I'm giving you an example. So, it will have a reaction with the oil, and then it can become a sensor. It can give us a like we have inserted the gene for the sensor, and when it is going to pick that oil, and when it is going to come in contact with the oil, it is going to give us the signal that the spilled oil is present here. Why spilled oil? Because there are many machines which are very very complex in the different for example factories, for example different fermenters. So we don't understand where the issue is, where the issue lies in the machine, and where is the oil getting leaked from. So we use biosensors, and then these biosensors can sense the spilled oil and give us a signal. As a result, they can glow and they can do anything, or they can produce a certain smell. So because of these signals, we can know that that wherever the fault in the machine is present. Understand? Fears are also being expressed about advances in the biotechnology in terms of release of harmful organisms. So we can get feared also, and we can get afraid also. Why? Because many different harmful like organisms can also be produced. For example, some disease causing organisms can be produced with the help of recombinant DNA technology. Now, fermentation is something which is very very positive, and which is so fermentation is actually something which is very very natural. And what happens is that there is cellular respiration of what, of the bacteria which we are going to add, and then glucose molecule goes through oxidation reduction reactions to release energy in the form of ATP. Fermentation is the process in which there is incomplete oxidation reduction of glucose, and that like it releases acid, and that we already know. Actually, this was implemented uh, in the branch of food in 1857. He convinced the scientific community that all the fermentations are the result of microbial activity. He showed that fertilization is always accompanied by development of microorganisms. So there is one microorganism, and then it divides, and then there are many microorganisms. For example, in case of the pizza dough, this is the agent is yeast, and when we are going to put the yeast, then the yeast divides, and then there is after if you check after 1 to 2 hours, there is gas inside the inside the dough. So the division of the microbe takes place, and then there are many kinds of fermentation, and each is a characteristic of a microbial group. So many see, that is how I explained you that it can be a bacteria and it can be yeast also. So different microbial groups are present. Fermentations are classified in terms and products form. The initial steps of carbohydrate fermentation are identical to those of respiration. So that is what we already know, that the first step will be the same, but the second step will be different. The process begins with glycolysis, and the glycolysis is the respiration, in which the glucose molecule is broken into two molecules of pyruvic acid, and but the second process is different. That is why different microorganisms proceed for the further reactions in different ways. It results in the formation of various products from pyruvic acid. Now pyruvic acid changes into various products. See here, what happens is that this is glucose. Because of glycolysis, we have pyruvic acid. The pyruvic acid can be converted into ethanol, which is a alcohol, lactic acid, and other organic acids also. So this is, for example, first kind of fermentation, second kind of fermentation, and third kind of fermentation. Okay, so sometimes we use this one and sometimes the other ones. Okay, now alcoholic fermentation is done with the help of yeast. And see, now three processes are described, and we are going to study two of them. Which two? The ethanol one and the lactic acid one. Okay, so what happens is that there are many types of yeast, and the The yeasts are known as Saccharomyces cerevisiae. This process is quite important and is used to produce bread, beer, wine, and distilled spirits. How is it produced? With the help of alcoholic fermentation. Okay, so the pyruvic acid was there, and then it is producing what? Acetaldehyde and carbon dioxide. This acetaldehyde is going to get converted into ethanol, and what we are going to get as a result? Bread, beer, wine, and distilled spirits with the help of alcoholic fermentation. In this process, carbon dioxide is removed from pyruvic acid. The product is acetaldehyde, and then it is reduced to ethanol. The carbon dioxide is the carbon dioxide causes rising of the bread. Understand? So this is what we get. We get um, alcoholic fermentation when the pyruvic acid changes to acetaldehyde and it is reduced to ethanol, and carbon dioxide is also produced. Now lactic acid fermentation is also there. In this process, pyruvic acid is reduced to lactic acid directly, and there is no production of carbon dioxide. And it is done by many bacteria, for example, Streptococcus and Lactobacillus also. So these both of them are the types and species of bacteria. It is quite important in the dairy industry where it is used for souring milk and for the production of various type of cheese. So this is how it takes place. Uh, we add certain bacteria and they are going to curdle the milk, and lactic acid is produced and fermentation is seen, and then different kind of cheeses are produced with the help of different kind of bacteria.